We are ready to go through the different levels of protein folding. So there are 4 levels of protein folding. Most proteins will go through the first 3, and then some proteins, if they're really complex, will go through the 4th as well. So let's start with the primary structure of a protein, or the first level of protein folding. Primary structure is pretty easy. This is just the unique sequence of amino acids that are strung together to form that chain that is the polypeptide. Let's take a look at this animation.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
And so I really like that animation because it shows you how the amino acids are different, how they're similar, right? This structure right here did not change for any of the amino acids. The only part that changed was that R group down on the very bottom. Okay, so primary structure is like spelling a really long word correctly in English, right? You have a whole bunch of letters you can pick from. You have to put them in the right order to make sure that that word actually makes sense. Same thing goes for the polypeptides in cells. If the cell does not put the correct amino acids in the correct sequence, the protein won't fold properly and it won't make any sense. It won't have any function in the cell. And then you might also be asking, well, what determines what order the amino acids are going to be put in when a protein is being built? Well, this is determined by the genetic information. So your genes are going to hold the information that the ribosomes are going to use to make sure that they put those amino acids in the correct sequence when they're stitching together these long polypeptide chains. So primary structure, think of it like spelling, right? Making sure that the amino acids are in the right sequence. So once you've got your strand of amino acids that we call the polypeptide, it's time now to start folding it. So now we get to the secondary structure of protein folding. This is found in most proteins, and it contains very specific structures, either alpha helices or beta pleated sheets. So let's check them out.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so secondary structure has 2 main shapes. Here you have an example of your alpha helix, where you've got essentially the polypeptide is just coiled, and then the beta pleated sheet is where the polypeptide goes in one direction, comes back, doubles back on itself, and then comes back again. And it's called a beta pleated sheet because it kind of has an accordion-like structure in that region. Now, in order to maintain these 2 different shapes in the secondary structure, you have to have some bonds there holding it in place. And this is very important to understand that secondary structure is held together by bonds between parts of the backbone. So that similar portion of every amino acid holds secondary structure together, and there's one type of bond that's used here. It's only hydrogen bonds between those backbone components that help to hold the alpha helix together or to help to hold the beta pleated sheet together. Once you have your alpha helices and your beta pleated sheets, then you can get to tertiary structure. And this is where you take those existing shapes and you kind of blend or mesh them all together to give the polypeptide its final shape. So let's take a look at this animation.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, so to review, tertiary structure is this final overall shape for one polypeptide. And in this case, different from secondary structure, tertiary structure is held together by interactions between the R groups, so those different parts of each amino acid. And we can use a variety of different interactions to help hold that shape in place. Interactions include pretty much every single bond type that we've talked about. So covalent bonds, van der Waals interactions, hydrogen bonds, ionic bonds, and also some hydrophobic interactions are involved there as well. And so this diagram gives you kind of a review of what was in the video. So the purple ribbon here is the abbreviated backbone of the polypeptide, and then we've highlighted select R groups here on different amino acids. So here's one R group, here's another R group on a different part of the polypeptide, and you can see there's a hydrogen bond here forming between those two R groups. Here you've got R groups that are primarily hydrocarbons. Right? Hydrocarbons are very hydrophobic. Right? There's no charge separation there, nothing to track the water. And so you can see that these R groups, this one right here, which is identical to the one on the bottom, they're clustering on the inside of the protein away from the water, the aqueous solution on the outside. They want to be protected. Here you have an example of a disulfide bridge. So this is a covalent bond between these two sulfur atoms. So this was an R group here, and the R group at the end had a sulfhydryl group. And then this is the same R group but on a different amino acid. Again, this one had a sulfhydryl group. The two sulfur atoms ditched their hydrogens, said peace out, and instead decided to form a covalent bond between themselves. And so that's a disulfide bridge. And this is an area of the protein where that tertiary structure is held really tightly in place. And then lastly, here we have an example of an ionic bond holding together parts of the protein. So this R group down here has an amino group, which remember can act as a base. And then here on this R group, we've got a carboxyl group. And this, again, remember, can act as an acid. And so when these two groups ionize, they develop charges. These two opposite charges are going to attract and form an ionic bond between the two R groups to maintain the shape of the polypeptide in that position. So those proteins that exist as just a single polypeptide will reach that tertiary structure, will be fully folded, and hopefully have their fully functional shape and can go on to do their business within the cell. Some proteins, however, are huge, and they actually are made out of multiple polypeptides. And these are the ones that are going to reach that quaternary structure of protein folding. This is where you have multiple polypeptides coming together, they each fold, and then they kind of aggregate together or fold together to form the very large protein. So let's take a look at this animation.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, The red oxygen-carrying protein of blood is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, so hemoglobin was the example of a multi-polypeptide protein that was used in that animation. You can see another rendition of that hemoglobin here. You can see that it has 2 alpha subunits in dark blue, 2 beta subunits in that teal color, and each one has that heme group with the iron in the middle. And this is what helps to attract the oxygen to the protein located in the red blood cells, so the red blood cells can carry that oxygen throughout your body. Now, hemoglobin is called a globular protein because, well, not very scientific, but it looks like a glob. It doesn't have a very, very distinct shape to it. Not all proteins that reach that quaternary level of protein folding are globular. There are also fibrous proteins. So collagen, what's found in your skin, for example, to help maintain that nice rigidity to your skin when you're young. Collagen is actually a fibrous protein, and it's made out of 3 polypeptides that sort of twist around each other to create this fiber, very much like a rope or twine or yarn, right? And it gives that structural integrity to that protein as well.